All right, guys. So mutations this week, or mutation repair. Last week was mutations. We talked about point mutations, frame shifts, and the kind of DNA damage that happens. So we're going to talk about that. We're going to talk about how we fix these problems. So we're basically going to focus on this region right here. How do we actually do DNA repair? So it's not just mutation repair; it's also DNA damage repair. So on the left are bacteria cells, on the right are eukaryotic cells. Basically, we have more tools at our disposal, but bacteria do have a lot of methods as well to repair their DNA. But basically, what you're trying to do is you're always weighing the pros and cons. You say, okay, I found some DNA damage. Maybe there's a substitution. Maybe I have a frame shift. Maybe I have a single-stranded break. Maybe I have a double-stranded break. Whatever it is. I need to weigh my options. So, how much energy, or i.e., what enzymes do I have to make? How much energy do I have to spend to actually fix this problem? Or is it easier to just have the cell die? And that's a good question. So when you do have your cell die, we call that apoptosis, okay? So bacteria have apoptosis-like processes, but generally we want apoptosis to occur in our cells because apoptosis does not cause inflammation. Necrosis, which is like just cells dying for whatever reason, like if they got stabbed or something and they're dying, that's necrosis, and that will cause inflammation. So in general, if your cell's not doing so great, maybe you got a big dose of radiation or whatever, can you fix it? And if not, then you need to destroy the cell because you don't want it to become cancerous because, cancer is bad. So let's get to it. So at the top here, we have are different damaging agents, okay? So we've talked about these before. This was all last week. So replication error, when your DNA polymerase makes a mistake. You can have reactive oxygen species. You can have chemicals get in there. You can have UV light. You can have different kinds of ionizing radiation, so like your X-rays and your gamma rays. And then you have at the bottom here the actual kind of damages that they cause. So maybe your bases are wrong. They're lined up wrong. You have a single-stranded break. You could have double-stranded breaks. Those are the worst. You could have agents change the nucleotides so that they look wrong or they're binding wrong. And UV light can cause pyrimidine dimers or thymine dimers. Now, we're actually going to talk a little bit at the end about chemotherapeutics, so stay tuned for that, because this isn't always necessarily the worst thing in the world. Maybe this is a cancer cell that we're trying to kill, and so we're going to use like radiation or chemotherapeutics to actually cause some of this damage in the hopes that we can actually, destroy the cell. But we're going to go through each of the ways that we fix these things. Now, there's really only 5 ways. There's subtleties, there's multiple versions, but we generally lump things into 5 different categories. And they're on the bottom here. So you have your mismatch repair, your base excision repair, and your nucleotide excision repair. And those are going to be the ones that make kind of the most sense. You're basically just going to take a chunk of nucleotide, either one nucleotide or a chunk of them, and then replace them. And so we also have some issues. So if you have a double-stranded break, we have special systems for dealing with that. And so that's what we'll talk about there. So let's get into it. Let's talk about replication error. So we talked about this last time. Your DNA polymerase can make mistakes when it's extending the actual DNA strand. So DNA polymerase is going through replication, right? It's making a new daughter strand. And sometimes it incorporates the incorrect nucleic acid. Man, I always say that. The incorrect nucleic acid. And if it does that, it can detect that. Now, how does it actually do that? Well, that's a really good question. I mentioned it last time. So basically what happens is you have 2 domains. Okay, so it's a big old protein, and you have 2 domains for that protein. Domains are areas of a protein that do something. So you can have one protein that has multiple domains, i.e. it has multiple functionalities. And so in this case, the polymerase activity, where it's actually making new nucleotides, or it's not making nucleotides, where it's adding new nucleotides to the DNA strand, that is one domain. But if it incorporates the incorrect nucleic acid, that causes a conformational change in the DNA, and that causes the strands to shift into a different domain. And that's the exonuclease site. Now, you can buy polymerases that don't have exonuclease sites. There are polymerases in nature that don't have exonuclease activity, is generally what it's called. But most of them that work inside of your body have these, and most creatures actually have exonuclease activity. Now, how good it is at its job, that's another question, but it does have some fact-checking, is usually what we call it, or some just back-checking. So it goes from, normally we're building from the 5' to 3' direction, right? We always build from 5 to 3. So that's what's happening here. So we're building from 5 to 3. So we're taking this new thymine right here, and we're sticking it on that 3' end. So we're building 5 to 3. We detect a problem, so we send our strand into the exonuclease region, right? So in this case, we put a thymine next to a guanine. That's wrong. And so we detect that, and we have 3 to 5' exonuclease activity. And so that's what this is called, 3 to 5' exonuclease activity. And that's just a way to fix that problem. Now, let's say you have a mistake that happens. You're going to fix most of the mistakes with this process, but you know what? Every now and then you're just going to accidentally incorporate one. Well, there's something interesting that happens. So when you make your new strand, what you do is, so the parent strand is on the bottom and the daughter strand, the one we just made, is on the top. And notice you methylate your parent strand, okay? And so by doing that, that lets us distinguish between the two strands. So now I can tell the methylated strand, so there's all these methyl groups on that backbone, that tells me that this was the parent strand and that this is the one that I should use as my template. This is the one that should, these are correct. And so the non-methylated strand is mismatched. Something is wrong. And so we use mismatch repair to actually fix that. There are a few enzymes that do that. They're all called mute enzyme. There's mute H, mute S, and mute L. And what they do is they just look for these mismatches. That's their job. They basically look for incorrect binding here. And if they detect that in the daughter strand, so they kind of move along that daughter strand because it doesn't have the methyl groups. And so they're like, okay, well, we know that this daughter strand doesn't have, this is the daughter strand, it doesn't have methyl groups. And so if we see some kind of problem, we know to go in and replace that chunk of DNA. And it will replace a few nucleotides, like 7 to 10-ish, just in that whole region. So it cuts out this whole little chunk here. Okay, so that's kind of cool. So it's not just one single base that's fixed, it's a little chunk of bases. Is mismatch repair. Now, mismatch repair is only able to happen because we have not yet methylated that new daughter strand. That actually happens very, very quickly after replication. And so you have to do your mismatch repair really, really quickly. And so let's say that you miss it. You missed out on one of the nucleotides. Now you can imagine you're actually fixing most nucleotide problems really, really quickly. And so it's pretty rare that they actually get through both these processes, the replication exonuclease activity and your mismatch repair. But let's say you did get one through there. Well, that's where base excision repair comes in. And so what we're looking for is a mistake that happened. Now, usually this is going to be because of damage, not necessarily because your polymerase made a mistake. Mismatch repair and your polymerase's exonuclease activity, that's because usually that's because your polymerase made a mistake. Okay, that's what those are for. If you have a mutagen, if you have like a chemical come in, or I guess you could argue UV light, but we have a separate thing for UV light. So like a chemical, okay? So either you modified a base to change it so it's not correct, or you have some kind of chemical that looks like another base, but regardless, you just have one side that's wrong, okay? So one single nucleotide is messed up. It could be a couple nucleotides, but we're kind of focusing on just one mistake. And so you use base excision repair to actually fix that, okay? And so I have the order of what's going on here because the next one, which is called nucleotide excision repair, follows kind of a similar functionality. So basically, we have a series of enzymes called glycosylases that look for incorrect bases. That's what they do. They kind of scan along and look for problems. And if they detect a problem, they actually take the nucleotide, okay, so the base part of it, not the backbone, but the actual nucleotide of it. So I put a little nucleotide on here. So here would be our sugar phosphate backbone down here, and then this is the base. And what it does, it kind of like flips it out and then cuts that glycosidic bond. And so there's the glycosidic bond between the sugar phosphate backbone and your actual base. And so it cuts the base out, which is really cool. It talks about, when you read about this, it actually literally like kind of bends the whole backbone out and flips that nucleotide out away from the hydrogen bonding, so it's easier to cleave. And so it cuts that. And that forms, it's called an abasic site. That's a better term for it. So they're also called AP sites. That stands for apyrimidine or apurine site, which abasic meaning no base. That's just the term of use. And so if it's abasic, it doesn't have a base anymore. It's abasic. If it's an apurine, it's an apyrimidine site, same thing. So this is just a more generic term. It can be a pyrimidine or purine, doesn't matter. And so the backbone is still there, and so we need another enzyme to come in called an AP endonuclease, and it looks for these AP sites. Okay, so we've gotten rid of the problem, right? So now, after getting rid of the problem, we've now got to fix the actual bonds that are there and replace the nucleotide that was removed. And so we'll use the other strand to actually do the replacement. And so that's the job of the endonucleases. So they look for these sites. So they don't care what was there because it could have been anything. So they just find the little hole that's there and they come in and they will cleave the backbone either in one little spot. So there are short patch AP endonucleases and there are long patch endonucleases. And so a short patch will just do one single nucleotide. Long patch ones will do 2 to 10. Which isn't that dissimilar from mismatch repair, but because it's like a little chunk. But you can have a single base repair or like a little region of nucleotides. Because sometimes, I don't know, you might just kind of assume like, hey, there was one mistake here. Let's just cut out a couple of these nucleotides just to make sure that we're kind of getting this whole little region fixed. So the endonucleases cleave that backbone, and then other enzymes will fix the ends of the backbone to make sure, because sometimes the endonucleases don't cleave at the correct carbon. And so it makes sure that those are all ready. And then a DNA polymerase, not the typical kind that would normally do the elongation, but most creatures, even prokaryotes and eukaryotes, have a special polymerase that will fill in these little gaps that happen. So like in prokaryotes, it's Pol 1, and eukaryotes, it's Pol beta, which those are not the normal ones that we use to actually extend our normal replication. Our normal DNA strands. And so then, so they fill in those gaps, and then you still got one little gap in that backbone, and that's where ligase comes in. And ligase will actually finish and rebuild that phosphodiester bond, and then you're fixed. Now, you can also imagine if you had a mistake like a single strand break, so instead of just having an incorrect nucleotide here, maybe this whole chunk here is broken, well, then just cut out the piece and replace it. And so that's base excision repair is also used to fix single-stranded breaks. Now, these mutations come from like a mutagen, like a chemical or something. Now, if you have UV light that causes damage, we as creatures have been dealing with UV light damage for as long as our ancestors, have existed. And so UV damage is really, really common. And so we have a special system that's kind of similar to that. The end mechanism is similar in that we're going to take a chunk of this, cut it out, and replace it. But it actually has a few different enzymes. We call that nucleotide excision repair. Now, I will say, it gets confusing, base excision repair versus nucleotide excision repair, okay? I understand that they sound similar. So the base one, that's when you have a chemical causing it. Nucleotide excision repair is when there's UV light damage. Now, we know that if UV light causes damage, what that does is it causes these things called adducts. And so if you have 2 thymines or a cytosine and a thymine or 2 cytosines stuck together, we know that that can cause a frame shift, right? It's really, really dangerous. We call those adducts, okay? So it's kind of like they're stuck together. As opposed to having just the incorrect nucleotide, we know that it looks wrong. There's these things stuck together. So we call those bulky adducts because they're like physically kind of bulky. They're this big old honking thing stuck on the actual DNA. And so that's really bad. And so we use 2 different ways to actually recognize that. Global genomic nucleotide excision repair just has special enzymes that look for dimers, basically, like thymine dimers. Now, what else you can do, so they're looking for it, but let's say they miss it, but let's say you're doing transcription, right? Your RNA polymerase comes along and it's chugging along, and it encounters one of these thymine dimers. Well, that's not good. It's going to hit it, and it's going to at least pause, if not fall off. And so we can detect that with a variety of enzymes, which there's a bunch right here. And so we call that transcription-coupled nucleotide excision repair. But it kind of makes sense. Like, you're supposed to be doing transcription, right? You're making messenger RNA off of a gene, which is DNA. And if there's a thymine dimer, your RNA polymerase can get kind of stuck on that little thymine dimer. Now, you do remove a larger portion, because if you got one area of thymine dimers or cytosine dimers or whatever, the chances are there's probably a lot of damage that's occurred in that region. And so we remove 25 to 30 nucleotides. So it's a bigger chunk than what we typically do for base excision repair, which maximum usually like 10 nucleotides. But the whole process, honestly, is relatively similar. Like you're going to cut it out with an endonuclease, and you're going to repair it with a DNA polymerase, and then you're going to religate it. So the actual process, that's why I spent some time talking about this process. The actual enzymes that you use, they might be different enzymes, but they have the same activity, and so that's why I included that. Now, as we keep on cruising, what happens if we have a double-stranded break? So double-stranded breaks are bad news, okay? Real bad news. And there's two big ways that we have to fix them, okay? Because we encounter them all the time. So you can have DNA damage occur all the time. So if you have a double-stranded break, you have two options, okay? If you are in the S or the G2 phase, okay, so of the cell cycle, so that means you have done your DNA replication, you have your sister chromatid, right? So you have a copy of each gene from mom, and you have a copy of each gene from dad, and when you go through the S phase, you double those, right? So you have 2 copies from mom and 2 copies from dad, and they're stuck together. So those are a sister chromatid. So if you have a sister chromatid sitting around, and one of those sister chromatids has a double strand break, well, basically what you do is you kind of hold it together, and then you use the other sister chromatid, because they should be identical, you use that sister chromatid to actually replace the lost region and make sure that you're doing it correctly. So that is called homologous recombination. Though there are a couple ways that actually happens that we're not going to go into, but basically you just use enzymes to replace that region. And so you can see here, the enzymes come along, so there's the broken strand, and then there's the strand that's fine. So these would be 2 sister chromatids, and they're probably attached to like a centromere or something. And so you can use that to help you replace and make sure that there are no mistakes. Now, you can do that with homologous chromosomes. So if you have 1 copy from mom and 1 copy from dad, you can, let's say the damage occurs in mom's chromatid or chromosome, you can use dad's to fix that, but that it doesn't always happen, because I mean, what are the odds? First off, you could have a mistake that way, because what if there's a mutation in dad or something? So now I've sort of like replaced a chunk of mom's gene with dad's gene, so that's not ideal. But also, they might not necessarily be next to each other, okay? And so because we have the rules of independent assortment. So there's nothing that says that the chromosome from mom and the chromosome from dad have to hang out next to each other if they're the same chromosome. So the odds of that actually happening are much more rare. You would much rather prefer to use a sister chromatid to do that. Now, if you do have the damage occur outside of that, what you can do is non-homologous. Okay, so we're not using homolog, so that would be like your sister chromatid would be homologous, or the chromosome from dad would be a homolog. So we can have non-homologous, okay, so it's not using any other piece of DNA, end joining. And basically what you're doing is you're taking advantage of the fact that there's probably not the loss of nucleotides. What's probably happened is your actual DNA backbone has been damaged. And hopefully what's happened is that you kind of have it cut in a couple different spots, so you have these overhangs. And so they'll kind of fit together like puzzle pieces, and you just need to religate those backbones. And so if it was a perfect right straight through those backbones and it's just kind of flopping around, then sure, it might be a little more difficult. But for the most part, you're going to have these compatible overhangs that will kind of stick together with those hydrogen bonds, and then you just religate. This happens a lot. Non-homologous end joining is super common. It's one of the main ways that you would do it. Now, there are some subtleties to it. There are different versions of non-homologous end joining. Some of them cause more mistakes than others. They can accidentally delete whole portions, which would be really bad. I mean, just in general, double-stranded breaks are really, really dangerous. And so fixing them is really, really important. But I just wanted to cover that non-homologous end joining is the more common way that we tend to fix these problems. Because what are the odds that you're actually in the S or G2 phase? Well, depending on the cell type, it could be super rare or never happen at all. So that's why this actually happens. Now, it does occur on purpose sometimes. And so I'm an immunologist, and so I wanted to talk a little bit just about how we pick the different regions when we do recombination to pick what portion of your antibody gets actually made. And so we have the variable region of antibody, that's the business end, the end that binds to stuff. And you have a bunch of different V segments, and you have a bunch of different D segments, and you have a bunch of different J segments. And what those do, so that's the variable, diversity, and joining, or the joint segments. And there's a bunch of different genes that code for those, and you want to pick one. So let's say there's 6 V genes. Well, you want to pick one of those 6. And let's say there's 20 D genes. Well, you want to pick one of those Ds. and then there's like 7J segments, and so you want to pick one of those J's. And so to actually pick individual chunks of those, we cut them out and then do non-homologous end joining to actually repair that problem. Okay, so but it's all kind of controlled. It's very controlled by these enzymes called RAG. And when I was in grad school, we had RAG negative mice. So the mice didn't have this, and so they couldn't do this. And so they would end up with B cells and T cells that didn't have antibodies or T cell receptors that looked right because they weren't able to do this, which is kind of cool. Now, I want to cut to a really, really good TED Ed video. I show this in like all the classes that I teach because it's just really good because it's simple enough that it addresses what we're talking about, but it goes into enough detail that it's actually super, super helpful. So let's cut to that.
The DNA in just one of your cells gets damaged tens of thousands of times per day. Multiply that by your body's 100 trillion or so cells, and you've got a quintillion DNA errors every day. And because DNA provides the blueprint for the proteins your cells need to function, damage causes serious problems, such as cancer. The errors come in different forms. Sometimes nucleotides, DNA's building blocks, get damaged. Other times, nucleotides get matched up incorrectly, causing mutations. And nicks in one or both strands can interfere with DNA replication, or even cause sections of DNA to get mixed up. Fortunately, your cells have ways of fixing most of these problems, most of the time. These repair pathways all rely on specialized enzymes. Different ones respond to different types of damage. One common error is base mismatches. Each nucleotide contains a base, and during DNA replication, the enzyme DNA polymerase is supposed to bring in the right partner to pair with every base on each template strand, adenine with thymine and guanine with cytosine. But about once every 100,000 additions, it makes a mistake. The enzyme catches most of these right away and cuts off a few nucleotides and replaces them with the correct ones. And just in case it missed a few, a second set of proteins comes behind it to check. If they find a mismatch, they cut out the incorrect nucleotide and replace it. This is called mismatch repair. Together, these two systems reduce the number of base mismatch errors to about 1 in 1 billion. But DNA can get damaged after replication too. Lots of different molecules can cause chemical changes to nucleotides. Some of these come from environmental exposure, like certain compounds in tobacco smoke. But others are molecules that are found in cells naturally, like hydrogen peroxide. Certain chemical changes are so common that they have specific enzymes assigned to reverse the damage. But the cell also has more general repair pathways. If just one base is damaged, it can usually be fixed by a process called base excision repair. One enzyme snips out the damaged base, and other enzymes come in to trim around the site and replace the nucleotides. UV light can cause damage that's a little harder to fix. Sometimes it causes two adjacent nucleotides to stick together, distorting the DNA's double helix shape. Damage like this requires a more complex process called nucleotide excision repair. A team of proteins removes a long strand of 24 or so nucleotides and replaces them with fresh ones. Very high-frequency radiation, like gamma rays and X-rays, cause a different kind of damage. They can actually sever one or both strands of the DNA backbone. Double-strand breaks are the most dangerous. Even one can cause cell death. The two most common pathways for repairing double-strand breaks are called homologous recombination and non-homologous end joining. Homologous recombination uses an undamaged section of similar DNA as a template. Enzymes interlace the damaged and undamaged strands, get them to exchange sequences of nucleotides, and finally fill in the missing gaps to end up with two complete double-stranded segments. Non-homologous end joining, on the other hand, doesn't rely on a template. Instead, a series of proteins trims off a few nucleotides and then fuses the broken ends back together. This process isn't as accurate. It can cause genes to get mixed up or moved around, but it's useful when sister DNA isn't available. Of course, changes to DNA aren't always bad. Beneficial mutations can allow a species to evolve. But most of the time, we want DNA to stay the same. Defects in DNA repair are associated with premature aging and many kinds of cancer. So if you're looking for a fountain of youth, it's already operating in your cells, billions and billions of times a day.
Gotta love those TED Ed videos, huh? Really, really good. Animated well, great info. It's awesome. So you just got to see behind the scenes. I have spots in the PowerPoint where I put the videos. So the last thing I wanted to talk about is just, there are two things. I wanted to talk about how much damage, how much repair actually occurs and how little mutations actually pass through to your offspring, and so, or to your, every time you replicate to the daughter cells. And so when you actually have error rates occur, you fix most of those with your polymerase activity and your proofreading activity. Polymerase selectivity would be like having your DNA polymerase have access to the correct nucleotides. How well does it actually grab the right nucleotide and put it in the right spot? Now that's different from its proofreading ability. And so you can have different selectivities, but for the most part, that's gonna fix a lot of problems. As long as your polymerase does a good job, awesome. And then you can proofread, so that's going to eliminate it down to 10 to the minus 6 possibilities. That's not a lot of mistakes. But I mean, as we've talked before, DNA is really, really long. There's tons of bases. And so if you're trying to replicate an entire genome, you could make some mistakes. And so that's where mismatch repair can really drop that down. And so this would be 1 out of a million. So this would be what, 1 out of every billion nucleotides. And so you can really start to fix some of these problems. And so when you actually have damage occur, I don't know, we fix it most of the time. We have all kinds of great ways to fix it. Now, the last thing I wanted to talk about was cancer. So if you get cancer, we got to treat it, right? So what are the ways that we treat it? Well, we use a variety of mechanisms, right? And a lot of those involve actually causing DNA damage. So either single-stranded breaks or double-stranded breaks, or using chemicals. So a lot of the chemotherapies that we use, like platinum chemotherapies, so that would be like last time, I can't remember the name of it, but in the last, cisplatin, that's what it was, is a platinum chemotherapy drug that people will use. And that blocks non-homologous end joining. It actually blocks PARP1, which I believe we've talked about before. And I think BRCA1. So it prevents non-homologous end joining, which we just talked a bunch about. And so if I can cause a double-stranded break using radiation or gamma radiation, X-rays or gamma radiation in someone's cancer, so you focus that super in, like using like a cyber knife or something, right on that little cancer tumor, and then I can also give them intravenous platinum chemotherapy, I can prevent the repair process. And then hopefully, I mean, that's why it makes you so sick, and it can really hurt your cells, but hopefully you kill the cancer if you direct those x-rays really, really well. And so I just wanted to kind of cover that there are lots of different drugs that we use that prevent what we've talked about, all of these repair pathways. And so that's just an important idea to keep in your mind, that just because you can have a repair pathway doesn't mean you always want that repair pathway to happen, like if you had cancer.